Welcome to The Explainer. Today we are diving deep into the science behind every single pill you take.
Have you ever really thought about it? Why one pill once a day, but another one every 4 hours on the dot? Well, you might be surprised to learn it's not a guess at all. It's not random. It's actually a precise science, and it determines the perfect dose for everything from a simple painkiller to a life-saving antibiotic. So let's break it all down.
To really get our heads around dosing, we first need to meet the two main characters in this story. These are the two core principles that pretty much govern everything a drug does inside your body.
So here's our roadmap for today. We're going to start with the basics, pharmacokinetics versus pharmacodynamics. Then we'll follow a brand new drug on its long journey to the market. After that, we'll see how it actually works on a tiny molecular level. And finally, we'll connect all of that science right back to what's sitting in your medicine cabinet.
Okay, let's dive right in. It all starts with these two fundamental and very different concepts, pharmacokinetics and pharmacodynamics. Honestly, these two ideas, they're the absolute foundation of all of modern pharmacology.
Here's a really simple way to remember the difference. Pharmacokinetics, or PK for short, is all about the journey the drug takes through your body. Pharmacodynamics, or PD, is what the drug actually does to your body once it gets to its destination.
So basically, PK is all about measuring the concentration of a drug in your system at any given moment. It's kind of like tracking a package, from the second it enters your body until it's been processed and finally removed. This tells scientists exactly how much of the drug is available to do its job and, just as importantly, for how long.
And on the other side of the coin, you've got PD. This is where the magic happens. It's all about the drug's actual power to create change. So, if you think of PK as the quantity of the drug in your system, PD is all about the quality of its effect.
And this is a really crucial point. We have to understand the difference between a response and an effect. So in this example, the blood pressure readings, the 92 and the 82, those are the responses. They're just direct measurements. But the effect, the thing that we really care about, is the calculated change from where we started. In this case, that's a drop of 10 points. That's the effect.
And being able to precisely measure an effect like that is exactly what scientists rely on when they take a new drug on its incredibly long and carefully mapped out journey, all the way from a lab discovery to your local pharmacy.
So take a look at this flowchart. Getting a drug to market is a massive, multi-year journey broken down into these very distinct phases. It's an incredibly long and rigorous process, guided by regulatory bodies, all to make sure a drug is both safe and effective. And that entire journey, from start to finish, is guided by this PK/PD modeling. It's like the compass they use to navigate from one phase to the next.
Each and every one of these phases uses PK/PD modeling to make absolutely critical decisions. From the preclinical animal studies that figure out the very first safe dose for a human, to the phase 1 safety trials, all the way to those huge phase 3 studies with thousands of patients to lock in the final dosing strategy. It's just this constant process of refinement.
Okay, so we've seen the big picture. Now let's zoom all the way in, like way in, to see what's actually happening at the molecular level when a drug really does its job.
It all starts with something called occupation theory, which is, it's basically a molecular handshake. A drug molecule, it's also called a ligand, finds and binds to a very specific protein in our cells called a receptor. You can think of it like a tiny molecular docking station. This binding, this handshake, it forms a complex that kicks off a whole cascade of events, and that ultimately leads to the effect we want.
But what's really fascinating is that not all of these handshakes are created equal. You can have different drugs that bind to the very same receptor, but they can produce wildly different results. And these curves right here, they tell that whole story. Some drugs create a really strong maximum effect, while others, they might do very little, or maybe even the complete opposite.
So a full agonist is like flipping a light switch all the way on. You get the maximum possible response. A partial agonist is like putting that switch on a dimmer. It turns it on, but only partway. Then you have an antagonist, which is so interesting. It binds to the receptor, but it does absolutely nothing. It just sits there, effectively blocking the spot so other molecules can't get in. And then there's the inverse agonist, which actually produces the opposite effect. It's just amazing. And this is how scientists can design drugs to have incredibly specific actions.
So, how does all this really complex science translate into the simple instructions on your prescription bottle? This is where things get really practical and, frankly, a little counterintuitive.
You'd think so, right? It seems totally logical. If you put 10 times the amount of gas in your car, you would absolutely expect to go 10 times as far. But when it comes to pharmacology, the relationship between the dose you take and how long the drug actually works, well, it's not nearly that simple.
In fact, according to the data, a massive 1,000% increase in dose, that's 10 times the original amount, might only give you a 50% longer duration of effect. Just 50%. That's it.
So why is that? Why doesn't it work the way we think it should? Well, it's because your body's elimination systems, we're talking mainly your liver and your kidneys, they work like a really dedicated cleanup crew that's on a fixed schedule. They can only clear out so much of the drug per hour. So piling on a massive dose just creates a backlog. It doesn't actually change the speed of the cleanup crew. If you really want to extend a drug's effect, you have to find a way to slow down that elimination process itself.
This very same science also helps us understand drug tolerance, that phenomenon where a drug seems to lose its effectiveness over time. And this can happen for a couple of key reasons. Either your body gets better and faster at eliminating the drug, that's PK tolerance, or your cell's receptors actually become less sensitive to it, and that's PD tolerance.
So, as you can see, understanding this really intricate relationship between dose, effect, and time is absolutely fundamental. It is the key, not just to healing people safely and effectively, but also for tackling some of the biggest challenges we face in medicine and in society, from managing chronic conditions all the way to understanding the very nature of addiction.